Okay, so these are cell cycle notes. As you can see here, it shows you, it starts off with kind of a definition, and it's talking about the cell cycle. It's just the life cycle of the cell, just this continuous process that the cell goes through during its life. This cycle includes three things: cell division, cell growth, and DNA replication. Each one of these kind of coincide with a major event or a major process in this life cycle of the cell. The first one, cell division, is really just known as, let me change the size of that, mitosis. That's where the DNA, cytoplasm, and organelles are divided up. So you start off with a single cell, then you end up with two cells, and they all have the DNA they need, they have the cytoplasm, and they have all the organelles that they need. Okay? So that's cell division. And cell growth is also known as interphase, and you've been learning about this already. But interphase is just the growth of the cell. It's the time in the cell's life where it's growing from its original daughter cell to the point where it can't grow anymore. So that's cell growth of interphase. The interphase has three stages, and we're going to look at them in a minute. And that's cell growth. And then we also have DNA replication, where DNA is copied. When you start off in the original cell, you have one copy. Now you have one copy of DNA and another copy of DNA. And this is so at the end of mitosis, when the two daughter cells are formed, each of those cells has a complete copy of DNA. So if we continue, we also have stages or events that occur during the cell cycle. The first one we've already talked about here, this one right here, cell growth, or also called interphase. And like I said, it has three parts to it. And some people even add one called G0. Talk about that in a minute. The G1, the S phase, and G2. G1 is your initial growth phase. And during this phase, it's at the end of mitosis. A new daughter cell has been formed, and then it just starts to grow. Produce new organelles, produce everything that it needs to be a viable cell. And that's G1. Then S phase is this part of this interphase where DNA is then copied. What happens is, during this initial growth phase, the cell reaches a size where it's no longer viable. It's reaching the point where it's no longer viable, and then there are certain triggers within the cell that tell it that you're getting too big, and you're going to need, eventually here, we're going to need to split into two cells. So because we're going to do that, you need to have a copy of your DNA ready. So as a response to all those chemical messengers and such in the cell, the DNA is copied, and that's the S phase, or synthesis phase. And that's your second phase. And then you also have G2 phase, which is the preparation for cell division. So once the DNA is copied, the cell is then ready to go through the next event, which is mitosis. But I was telling you before, we also have this G0 phase. G0 phase is where is the part of interphase where the cell kind of goes out of A, B, or C. It stops growing, it doesn't copy DNA, and it doesn't prepare for cell division. An example of a cell that does that are nerve cells, or neurons. Okay. They get to a certain place, and they don't produce new cells anymore. They just stop. And you've probably heard in biology classes, your brain reaches a point where it's not going to grow anymore, and it becomes more difficult for you to learn things and so on. So it's important, as a student, that you take advantage of the time that you're in right now, because your nerve cells are going to reach a point and they're not going to continue to grow and make new connections. connections in your brain, and things are going to be a little bit harder to learn. But that's called the G0 phase. Not all cells go through that, but some do. Okay, it's just like a continuous interphase. Never goes through mitosis or cytokinesis. It just stays in interphase. So that's your first event. Next event is cell division. Cell division is also known as mitosis. Put that down. Mitosis. Erase some of these other things. Okay. So mitosis, or cell division, has 4 phases. And they are prophase and metaphase are the first two phases. During prophase, this is just your initial phase. So, and really, what the main thing to remember is that the most important of this is that at the end, the new daughter cells have the right amount of DNA. So it's all about the DNA, these phases. It's all about the DNA. And really, if you understand that, you can kind of look for the main things that are occurring in each one of these phases. Prophase is exposing DNA. That's really what's happening during prophase. Because before this, DNA is located in the nucleus. And if it's in the nucleus, it can't leave. It can't get out. So what happens is, during prophase, the nuclear envelope breaks down. NE breaks down. So now you have these copies, 2 copies of DNA, and they're just floating in the cytoplasm. And they're ready for the next phase, metaphase. But also what happens is you have these things called centrioles. Okay? And these are kind of like guides. Guides for the DNA. And they kind of attach to them. You know, they're like a really good bodyguard. And they attach to specific DNA or chromosomes, and they move them around. And they do this by producing spindle fibers, which attach to each of the copied chromosomes. The prophase, exposing DNA, and the centrioles form and form spindle fibers and attach to the DNA. Okay. And during metaphase, you have the centrioles align the chromosomes in the middle of the cell. So what you have, it kind of looks like this. You have a cell. You have this region in the middle. It's called the metaphase plate. And the centrioles are in the corner here, opposite each other. It's not very good pictures, but they're good enough. And you have these DNA. They're all lined up in the center, depending on how many chromosomes you have. And then these produce little spindle fibers that are attached to each DNA. Each one of these do that. And this right here, this little X, is just a copy. There's one copy of DNA, and there's the other copy. And they're going to get pulled. One's going to get pulled that way. One's going to get pulled the other way by the spindle fiber. But these centrioles produce these spindle fibers. They attach to the chromosomes and they line them up. It's kind of like the same idea of organization. If you're a teacher and you have a bunch of little first and second graders, and you want them to go to lunch and do other certain specific things, you usually don't have them randomly do it. Because if you do, it'll be chaos. And maybe one of the little first or second graders will get hurt. Or some of the jobs won't get done that need to get done. Or maybe they'll end up in places they shouldn't be. So centrioles are kind of like keeping order. So they align all the DNA up in the center. So that when it is eventually pulled to each opposite end, it's done in an orderly fashion. And things don't get mixed up. You don't end up with too much DNA on one side and not enough on the other. But that's prophase and metaphase, your first 2 phases. Let's go to the next ones. Then, get this. Come on. Put this here. Okay. So we have anaphase. And telophase is your telophase, is your next two phases of metaphase. And anaphase is where the DNA gets pulled to opposite ends. This all has to do with the centrioles and the chromosomes. It's really the main thing that's occurring. The DNA gets moved around, split up, then it gets pulled to opposite ends. Anaphase, once it's lined up at the metaphase plate, the centrioles then start pulling it to opposite ends. Then we have telophase. Make it pink, purple or something here. What happens in telophase is DNA is now in opposite ends of the cell. The centrioles break down the spindle fibers, I'll say SF, and then the nuclear envelope reforms. Okay. Kind of looks like this. You have a cell, like this, and you have a nucleus on one side and a nucleus on the other. There's DNA in here now, okay. And you have the nuclear envelope reforms. So now you don't have this DNA free floating anymore. It's contained, it's protected, and it's ready for the last event, and that's cytokinesis. And also the centrioles then pull their spindle fibers in, they get broken down, and then they're not involved anymore. Cytokinesis is the splitting of the cell. Splitting of the cell. And it forms two new cells now. And these are called daughter cells. Okay. What happens is something called a cleavage furrow forms in the middle of the cell. Kind of starts looking like this. Bend in of the cell. And it splits right down the middle. And you have one cell here. And you have another cell there. And you have two new cells. And then the next process is interphase and cell growth. Now, you're going to learn next couple units about problems that can occur. A couple problems that you're going to learn about is the idea that most of the time centrioles work good and they pull the correct number of chromosomes to each side. Or every once in a while something happens. Maybe the chromosomes don't separate like they should, or maybe one spindle fiber pulls all the chromosomes. It's original and it's copied on one side, and one side doesn't get one part of the DNA. And that ends up in genetic disorder, or it can end up just death to the cell in general. And also what can happen is a cell, because if you remember, there's events. There's interphase, mitosis, and then cytokinesis, then it goes back to interphase. Well, some cells, though, they don't go back to interphase. They just keep splitting and splitting and splitting. Uncontrolled cell growth. That's cancer. We're going to learn about those in the next 2 units. And kind of interesting. I hope you enjoy those little videos with those people talking about what they think about laws, about cancer and such, and make sure you take your time there. And if you have any questions, send me an email, and I hope you're enjoying the notes and such in the class. And if you have any questions, just let me know.